That too, and then I have to go pack it up.
Of course.
Actually, really good.
Good Monday morning. This is our last week together. Next week, Dr. Cummings starts for the last 4 lectures. You have Dr. Cummings again. All of the very last lecture, which I think is April 2nd, the last lecture might be a review lecture. So depending on how his lectures go, the last lecture might be material or it might be a review lecture, depending on, like I said, on Dr. Cummings' last 4 lectures. All right, so you guys should have gotten your results back for test 4 as it's written in the syllabus. Can you please wait 24 hours before peppering you with questions about the test? So you think about them and, look at the questions and stuff before coming to me. So wait 24 hours before asking me about test 4, please. With the completion of test 4, that now allows us to kind of look at people who need to write the makeup exam. So this is only for students that have missed 2 or more tests. Right, there's only a couple dozen of you that are in that situation, but if you've missed 2 or more tests, and if you have appropriate documentation, so you have to submit undocumented or documented SAPs, and the instructions are sort of in the syllabus and up here as well as in an announcement I send out. So if you are writing the makeup will likely be this Friday, sort of the same time frame from noon till 10 p.m., and it's going to be same idea, 20 questions in an hour, except now the questions are cumulative. They cover the whole course. Okay, and that's because you've missed material from various places. You've missed at least 2 tests. Having said it's cumulative, at least it helps you study for the final, because the final is also cumulative. Okay, and we'll talk more about the final probably that last day, but hopefully it'll be a review. Okay, so let's get on to material that will be on the final. So last time we talked about types of membrane proteins. We talked about integral membrane proteins, lipid-linked proteins, and peripheral membrane proteins. Now we're going to see how those proteins get there, particularly the integral membrane proteins. How do you get a membrane, how do you get a protein into a membrane? Okay, so this is an overview slide. There's a lot of material on this, so let's kind of go over some of this. Okay, so now we're talking about proteins that have part of their structure within the membrane. So part of their protein is integral into the membrane. They have a part of the protein is spanning the membrane. And there's different ways you can do this. Okay, so you're going to have a transmembrane domain, basically. You can see at the end over here you have GPI-linked proteins. GPI are lipid-linked, but they actually start off with a transmembrane domain, and you'll see how that works in a second. So all of these proteins have a part of the protein that's inserted into the membrane. There are different classes, as you can see up here, different types, type 1, 2, 3, 4, and a variety of other things, and we'll talk about these. So we'll go through all these, and you don't need to memorize any of these types of proteins here, but you do need to memorize sort of their orientations. And we'll go through it. For example, a type 1 transmembrane protein always has the N-terminal domains that's luminal. It's always sort of outside of the cytoplasm. So when we go through all these, if I tell you something is a type 3 protein, then you know a type 3 protein has a carboxy-terminal domain in the cytosol, for example. So we'll go through these and hopefully that'll become clear. From this also, though, look at the membrane and look at the orientation. Even the textbooks flips things around all the time depending on how they kind of draw things. So you're going to have a cytosol side and an ectoplasmic side. So cytosolic and basically extracellular. Okay? They could be flipped in different diagrams, and they are. The textbook does it. I will do it on the exam. Make sure you orient yourself correctly. You know which is the cytosol, which is the luminal side. Right? Cytosol is always cytosol. Luminal is going to be in the ER, in the Golgi, in some sort of vesicle, or outside of the cell. Luminal is basically extracellular. So get those orientations right. The other thing to remember is that translation always occurs in the cytosol. Okay, ribosomes are in the cytosol. tRNAs are in the cytosol. The amino acids are in the cytosol. So translation is occurring in the cytosol. So the ribosome has to get the protein out of the cytosol into the membrane and into the luminal space. So if nothing else, you know the ribosome is in the cytosol, the protein starts off in the cytosol. It's got to be inserted into the membrane or into the lumen. Make sense? Okay, so translation starts in the cytosol. Translation starts, the ribosome is going to bind to RNA, and then translation is going to occur. As translation occurs, you're going to get this growing peptide, you're going to get this growing chain of amino acids. That ribosome is going to keep on translating unless it has a reason not to translate. If it keeps on translating and it gets to the end of the protein and nothing happens, that protein is likely a cytosolic protein. The ribosome translated everything, made a nice protein, the protein is going to stay in the cytosol likely. It can be targeted elsewhere, but for the membrane, to get into the membrane, it needs to get some sort of signal. And the signals that tell the ribosome to go to the membrane, go to the ER membrane specifically, are things called topogenic sequences. So if it's going to be a membrane protein, it needs a topogenic sequence. Topogenic comes from topology. Topology is basically shape. So there are certain amino acids that give the protein certain shape. And the ribosome recognizes this and knows, hey, this is going to the ER membrane. So these shapes are usually hydrophobic amino acids. Okay, so these are hydrophobic amino acids. When the ribosome is translating this, it makes these hydrophobic amino acids. It's sitting in the cytosol. They're hydrophobic amino acids. They don't like sitting in the cytosol because they're hydrophobic. They fold up into a shape. That shape, that topogenic shape, is recognized by the ribosome and then the ribosome transports that protein to the ER. So it's not to say that there aren't specific sequences. So these topogenic sequences are certain amino acids, and there is some sort of consensus in the types of sequences that they are. But more generally, these are hydrophobic amino acids. They form specific shapes because they're hydrophobic and they're sitting in the cytosol, and the ribosome recognizes that. Making sense? Okay, so these hydrophobic topogenic sequences can be kind of classified into 4 different categories. One of them you've already heard of before, without really necessarily calling it a topogenic sequence, and that's the N-terminal signal sequence. So you know that proteins that are going to be going to the ER, that are going to go to a translocon, have this N-terminal signal sequence. And that is indeed one of the ways you get things into the ER, or you get things in the membrane. So an N-terminal signal sequence, as the name suggests, as in the N-terminal domain of the protein, it's the N-terminal end of the protein. Signal, anytime you see the word signal, signal to what? Signal to go to the ER. So whether it's an N-terminal signal sequence or something called a signal anchor, it's signal, go to the ER. Okay, so the N-terminal signal sequence, you know from other things, it's going to be recognized and it's going to be taken to the translocon. And you're going to start translation in the ER, and we'll talk about that in a second. Okay, so that's one type of topogenic sequence. Another one is called a stop transfer anchor sequence, or STA. Okay, stop transfer anchor, it kind of tells you what's going on. You're going to stop transferring and you're going to form an anchor. Transferring into what? Transferring into the lumen. So the STA sequence is you're already on the ribosome, you're translating into the lumen, you're transferring protein into the lumen. The stop transfer anchor basically says stop transferring into the lumen, and then the rest of the protein starts translating back in the cytosol. And you'll see how that works in a second. Okay, so you have an N-terminal signal, you have a stop transfer anchor, you have something else called a signal anchor. This is an internal sequence in the protein, and it basically tells the ribosome tells the ribosome go to the ER. It's a signal to go to the ER. And you'll see how that works in a second. And the last one is something called the hydrophobic C-terminus. So the carboxy terminus of the protein, which is the last part of the protein that's being translated, is hydrophobic. So the ribosome is translating this protein, that's happily making this protein, and suddenly, just as it gets to the end, it reaches and it's got this hydrophobic thing. It realizes, well, this is hydrophobic, it's not going to be very happy in the cytosol, so this protein then has to be taken to the ER membrane. And it's done in a slightly different way, and that's exactly what we're going to start off with now. So we're going to look through all of these one at a time, starting with this tail-anchored protein that has this hydrophobic carboxy terminal domain. Okay, so again, translations occurring in the cytosol. The ribosome is in the cytosol. The ribosome is making a protein. It's going to translate this protein until it has a reason not to. In this case, basically, the protein is translated and has this hydrophobic carboxy terminal tail. So just as the ribosome is finishing this protein, it realizes, hey, this thing has a hydrophobic tail. It kind of coordinates things. So as the ribosome is releasing this protein, other proteins approach it and bind this hydrophobic tail. Okay, this hydrophobic tail doesn't want to be in the cytoplasm. So as soon as the ribosome finishes it, other proteins come and bind to it and take it to the ER membrane. Okay, so the first protein that binds to it is called GET3. So GET3 binds to this hydrophobic carboxy tail and then transports it to the ER membrane where there's 2 other proteins called GET1 and GET2. So then GET1, GET2, and GET3 then use ATP hydrolysis to insert this hydrophobic carboxy terminus into the ER membrane. So what you end up with is a protein that has a transmembrane domain, or has a domain within the membrane. That was a hydrophobic carboxy terminal domain. And then it has the rest of the protein is in the cytosol. So this protein has no extracellular domain. It just has a cytosolic domain and a transmembrane domain, which was the N-terminal hydrophobic carboxy terminal domain. Okay, so hopefully that's easy to picture. Ribosome makes this protein, the carboxy terminal end, the last part that's translated, is hydrophobic. It's recognized by GET3. It takes it to the ER membrane where there's GET1 and 2. So GET1, 2, and 3 together with ATP hydrolysis insert that carboxy terminal domain into the membrane. So you know that the N-terminal domain is in the cytosol. So the N-terminal domain of these tail-anchored proteins are in the cytosol. The C-terminal domain is in the membrane. There is no extracellular domain. Hopefully relatively easy. Okay, so now let's talk about these other types of proteins. The first one that's probably familiar to you, is one that's a type 1 transmembrane protein. Okay, this is the one that has an N-terminal signal sequence. So the N-terminal signal sequence, lots of proteins have this. That signal tells the ribosome you're going to the ER. So this is sort of showing you sequentially what's happening. So I'm not sure why they have this continuous mRNA here. Think of this as different time points. Right? The ribosome binds onto some mRNA. That mRNA then begins to be translated. Right? You're making protein. With the N-terminal signal sequence, the first thing that you translate is an N-terminal signal. So right at the beginning, right at the N-terminal domain, you translated this thing. It's a topogenic sequence. It's hydrophobic. It's going to be recognized by things. There's a signal recognition particle that Dr. Cumming may have talked about, but I'm not necessarily going to mention it, although I just did. There's a hydrophobic region. It is going to be taken. The ribosome will take it to a translocon. So the translocons are sitting on the ER membrane. Right? So the N-terminal signal sequence, which was a topogenic sequence that was recognized, is then going to be taken to this translocon, and it's going to be inserted through the translocon into the lumen. So the N-terminal domain of the protein is going to be in the lumen. Soon as that signal sequence is sort of translated into the lumen, it's cleaned off. So there's a signal peptidase that cleans off that signal sequence. So the signal sequence is not part of the mature protein. That topogenic sequence is not part of the mature protein. Okay, so the ribosome took this protein to the translocon because of the signal sequence. The signal sequence got put into the translocon, and then as it was translated into the ER lumen, that signal sequence is cleaved off. But the ribosome now keeps on translating. It's going to translate, and the protein is going to grow, and the protein is going to grow with the N-terminal domain in the ER lumen. If this was not a transmembrane protein, if this was an ER protein, the ribosome would keep on translating, and the whole protein would just end up in the lumen. And you can have luminal proteins that might be secreted or might be going to the Golgi or wherever. However, we're talking transmembrane proteins here, type 1 transmembrane proteins. These proteins have an additional topogenic sequence. These proteins have a stop transfer anchor sequence. So as the ribosome is translating this protein, it gets to this topogenic sequence, this hydrophobic region, this stop transfer anchor sequence. It then tells the ribosome and the translocon basically stop transferring to the ER, use this as an anchor. This is going to become the transmembrane domain. So stop transferring. If you stop transferring, translation continues in the cytosolic side. So as it's no longer transferring the protein, the ribosome finishes translation, and the carboxy terminal domain is in the cytosol. Okay, so you had two topogenic sequences, one at the N-terminal domain that got it to the ribosome. That's cleaved off. The N-terminal domain is luminal. Translation keeps on going until you get to a stop transfer anchor. That stop transfer anchor becomes the transmembrane domain. Translation continues, and the carboxy terminal domain is always in the cytosol. So all type 1 transmembrane proteins have an N-terminal domain that's in the lumen, and a carboxy terminal domain that's in the cytosol, and have one transmembrane domain, which was the stop transfer anchor sequence. Make sense? In Achieve, in a folder that's called non-testable material, there's several movies that try to explain how ribosomes go to the ER and how ribosomes put in secreted proteins and a variety of other stuff, including transmembrane proteins. So there's movies there and little tests associated with it, but those are not testable. So in Achieve, in this folder called non-testable videos, there are several videos of stuff like this. Although hopefully it's straightforward because my explanation was just that good. Okay, so that's a type 1 transmembrane protein. Built off of a type 1 transmembrane protein are GPI anchored proteins. Okay, so GPI anchored protein start off that sort of look like a type 1 transmembrane protein, but here they have a carboxy terminal STA. So they have a carboxy terminal stop transfer anchor. So a GPI anchor protein, or a protein that will be a GPI anchor protein, is like a type 1 protein. It's going to have an N-terminal signal sequence. It's going to get taken to the ER. The N-terminal domain will end up in the lumen. The ribosome will keep on going. But right at the very end, right at the carboxy terminal domain, that's where you're going to have the stop transfer anchor sequence. So the stop transfer anchor sequence is right at the carboxy terminal domain. So you've sort of made a type 1 transmembrane protein, except there's nothing in the cytosol. More important than that, if you're going to be a GPI anchor protein, there are certain sequences present in the protein right next to this transmembrane domain. These sequences are recognized by a protein called GPI transamidase. Okay? This enzyme is going to take this luminal part of the protein and cleave it off, and take this luminal part of the protein and link it to glycosyl phosphatidylinositol. Okay? So you don't have to memorize that. It's just GPI. And it doesn't look any different. I don't know why they draw these things like it's just another phospholipid in the bilayer. Okay, so what are you doing? You've made this type 1 protein, sort of, except that it doesn't have a cytosolic domain. You're taking the luminal domain and this enzyme, which you do need to remember for GPI transamidase, and you're taking this protein and linking it to an existing phospholipid in the bilayer. So now you have this protein that's lipid linked. It doesn't have a transmembrane domain anymore. You've cut off the transmembrane domain. It's just lipid linked. The protein isn't bound to the cytoskeleton and the cytosol. This protein isn't bound to anything in the extracellular matrix. So this protein, as are all GPI linked proteins, has very good mobility in the membrane. It can move laterally quite easily. Okay, so that's GPI anchored. Starts off sort of like a type 1 transmembrane protein, except its stop transfer anchor sequence is right at the carboxy terminal domain. And that actually stays in the membrane. The rest of the luminal part of the protein is going to be attached to a phospholipid through this GPI transamidase. And again, because this is luminal, once you get to the membrane, this protein is extracellular. It's sitting on the outside. All right, let's get some more complications in there. So that's sort of type 1, pretty straightforward. Type 2 and 3, sort of opposite versions of each other. Translations occurring in the cytosol, ribosomes in the cytosol, mRNAs in the cytosol, amino acids are in the cytosol. Translation starts up in the cytosol. The ribosome starts translating. It translates the N-terminal domain of the protein first. It's happily translating along, and suddenly it gets to a topogenic sequence. Okay, so it's already made an N-terminal domain, and then it gets to a topogenic sequence. That topogenic sequence is this topogenic sequence called a signal anchor. As the name suggests, it means signal, go to the ER. So it's going to take this protein and go to the ER. This topogenic sequence is hydrophobic. It's going to become the transmembrane part of the protein. That's the anchor part of it. The question is, you've already made this N-terminal domain. You already have part of this protein made. What do you do with this N-terminal domain? How do you orient this protein? Do you put this N-terminal domain in the lumen, or do you keep the N-terminal domain in the cytosol? Okay, so you can orient it 2 different ways. You've already made this part of it's going to be a transmembrane sequence. what do you do with this protein? What orientation is it going to be in the ER membrane? And that's the difference between type 2 and 3. You can see in type 2 the N-terminal domain is in the cytosol, and type 3 the N-terminal domain is in the lumen. Okay, so even if your mind, you can't picture how the ribosome folds the protein to do this, and you'll see when we get to type 4 that gets even more complicated. Even if you can't picture how the ribosome does that, it's okay, you just have to memorize that the type 2, the N-terminal domain is in the cytosol, type 3, the N-terminal domain is in the lumen. How does the ribosome do it? Well, the ribosome uses charged amino acids that are next to this topogenic sequence. Next to this signal anchor are going to be charged amino acids that will tell the ribosome what orientation to do it, to put it in. Charged amino acids are going to stay in the cytosol, is the simplest way you can look at it. So if a ribosome is making a protein, it's made the N-terminal domain, and as it gets to the N-terminal domain, there's suddenly a topogenic sequence. The ribosome then looks what is on the N-terminal domain side of that topogenic sequence. So in this example, here's the N-terminal domain. As it's translating, it gets to this topogenic sequence, this signal anchor. What's on the next to the signal anchor? Are there charged amino acids? There are. Charged amino acids are then, that domain is then going to stay into the N-terminal, is going to stay in the cytosol. If you remember when we talked about transmembrane proteins, integral membrane proteins, there are always charged arginines and lysines present right next to the plasma membrane. That's what these proteins are, these amino acids are. These charged amino acids that are telling the ribosome, this domain, the N-terminal domain that you just made, it's going to stay in the cytosol. When you continue translation, put the rest of it in the lumen. So if there's charged amino acids next to the topogenic sequence on the N-terminal side, the N-terminal side stays in the lumen. If there are no charged amino acids between the N-terminal side and the signal anchor, the N-terminal side gets inserted into the lumen. Isomatrix stays in the lumen, stays in the cytosol. Okay? So if there's a ribosome translating and it does the N-terminal domain, then it gets to the signal anchor. What does it do with this N-terminal domain? There are no charged amino acids between the N-terminal domain and the topogenic sequence, so the N-terminal domain gets put into the lumen, gets put through the translocation. As translation continues, actually you're now going to get charged amino acid on the cytosolic side, kind of confirming for the ribosome you made the right choice. Now there's going to be charged amino acids on the luminal side, not on the luminal side, to the plasmic side, my apologies. Therefore, this is going to stay in the cytosol. We've already mentioned that most transmembrane proteins have these charged amino acids on the cytosolic side, and that's the case here. Whether you're a type 2 or a type 3, you have charged amino acids on the cytosolic side, and depending on how translation occurred, with the type 2 you have the carboxy terminal in the lumen, with the type 3 you have the N-terminal in the lumen. I know I misspoke lumen at least once in there, but hopefully you get the pattern. And we'll go through this again in a second anyway. Okay, so type 1, 2, 3, they all have single pass. They all have one transmembrane domain. Proteins can have multiple transmembrane domains, and those are the type 4. Okay, so you need a signal. You always need a signal to get to the ER. So you need an N-terminal signal sequence, or you need a signal anchor. So far the ones we've been talking about are kind of have one or the other. With the type 4 now, it's going to get more complex because you're going to have multiple transmembrane domains, and the N-terminal domain can be on the luminal or cytosolic side, and the carboxy terminal domain can be luminal or cytosolic, depending on how many transmembrane domains you have. So it gets more complicated. Okay, and we'll go through that in a second. So the textbook sort of has this explanation of things. Okay, and we'll go through this, and then we'll go through things one at a time, and then go through an older version of this, which I think is easier to follow. Right, so we're looking at the topogenic sequences associated with transmembrane proteins. So the topogenic sequences are going to be hydrophobic regions that the ribosome is going to recognize, and it's telling the ribosome what to do anything. So the type 1
has two topogenic sequences, an N-terminal signal sequence and this stop transfer anchor sequence. Type 1 is always going to have the N-terminal domain in the lumen and the carboxy-terminal domain in the cytosol. Right? That N-terminal signal sequence is going to be cleaved off. So this signal gets to the ER, it's going to start translating, the stop transfer will become the transmembrane domain, and the rest of it goes into the cytosol. Hopefully pretty straightforward. Type 2 and 3, you're translating in the cytosol. Ribosomes are always in the cytosol. It's going to translate this N-terminal domain. It's happy to keep on translating. Suddenly it's going to get to this signal anchor. What does it do? This signal anchor has to be the transmembrane domain. It already made an N-terminal domain. It all depends on the charged amino acids around this topogenic sequence. If the charged amino acids tell it, keep that domain in the cytosol. So if the N-terminal domain is where the charged amino acids are, this domain stays in the cytosol. If there are no charged amino acids here, this goes into the lumen, and the other domain is in the cytosol. So those charged amino acids kind of tell the ribosome what stays in the cytosol. So far, so good? Okay, then you get into the type 4s. Okay, so type 4s, you're going to have multiple topogenic sequences, and you can start either in the cytosol or in the lumen, depending on the charges. The pattern is the same. Okay, the ribosome is always in the cytosol. The ribosome is translating in the cytosol, so it needs signals. If you see a signal, it means you were in the cytosol, you're going to the ER. If you see a stop transfer anchor, it means you are translating into the lumen. Stop that, go back to the cytosol. So signal means go to the ER. Stop transfer anchor basically means go back to the cytosol. Right? And then you get this sort of pattern of the protein being luminal cytosolic because it has loops going through. And we'll get to that in a second. Okay, so the textbook has chosen to kind of do it this way, which I find confusing, but I'm going to kind of back up a bit and kind of just remind you of what I just said, actually. Okay, so type 1, straightforward. Hopefully, as soon as you see an N-terminal signal sequence, you know that the N-terminal domain will be cytosol, will be luminal, sorry. N-terminal domain, luminal for type 1, always. For type 2, where you have this signal anchor, I should say, it all depends on those charged amino acids. Those charged amino acids are going to stay in the cytosol. So type 2, the N-terminal domain stays in the cytosol because of the charged amino acids. With type 3, the N-terminal domain gets put in the lumen, and then charged amino acids are translated, they're going to be in the cytosol. So far, so good? Okay. So let's get to the type 4A and B. Okay, so this is sort of an older version of the textbook where they have things labeled a little bit differently, but they've sort of labeled SA and STAs and sort of charged amino acids. Okay, so even if you can't picture what is happening, hopefully you can recognize the differences between the type 4A and the type 4B. Okay, a couple things. One is you can have different numbers of transmembrane domains. You can have different numbers of topogenic sequences. So in this example, this type 4A protein has 4 transmembrane domains. This type 4B has 7 transmembrane domains. So you can have, proteins can have different number of transmembrane domains, and that's related to the topogenic sequences. They can start off with the N-terminal domain in the cytosol, or they can start off with the N-terminal domain in the lumen. Make sense? As with the type 2 and 3. So, with the types 4A, the N-terminal domain is in the cytosol. With the type 4B, the N-terminal domain is in the lumen. Okay, ribosome is translating, starts off in the cytosol, it makes this N-terminal domain, it chugs along, and suddenly it hits the topogenic sequence. It's a signal anchor. Okay, so what does that mean? That means this is going to become a transmembrane domain. Ribosome, good, go to the ER. This is going to be the transmembrane domain. Wait, I've already made an N-terminal signal sequence. I've already made an N-terminal peptide. I've already made this N-terminal domain. What do I do with this N-terminal domain? This is going to be a transmembrane domain. What do I do with the N-terminal domain? It depends on the charged amino acids. If there are charged amino acids, this N-terminal domain stays in the cytosol. If there are not charged amino acids, the N-terminal domain is going to be put into the lumen. Okay, and then the ribosome keeps on translating. With the type 4A, it then kind of becomes sort of easy. You're in the cytosol, you put this in, this stays in the cytosol, my apologies. This stays in the cytosol, so now you're translating into the lumen. Stop transferring to the lumen, go back into the cytosol. Then there's a signal anchor, go back to the ER. There's charged amino acids that tell you that this stays in the cytosol. You're back in the lumen, and then you stop transferring, you're back in the cytosol. So you're always switching between lumen, cytosol, and there's usually positive amino acids that tell you you're staying in the cytosol. Right? So you're going to be switching between signal to go to the ER and stop transfer, which means you go back to the cytosol. If you can't figure out how the ribosome is doing this, it doesn't matter, just recognize the patterns. Okay, the type 4B is a little more complicated because you have this signal anchor, which means go to the ER, and this is going to become a transmembrane domain. Because there are no charged amino acids here, this luminal domain is going to, because there's no charged amino acids, this is going to become luminal. So this is going to be shoved through the translocon into the ER, and this signal anchor is going to be oriented, so now translation is going to occur into the cytosol. So you're translating in the cytosol, so if you want to go back to the membrane, you need another signal. Okay, so you have this double signal here because of the way the orientation is. If you can't picture that, it doesn't matter. If you see two signal anchors, you know it's a type 4. Okay? 4B. The type 4A starts off with one signal anchor, and then it goes signal anchor, stop transfer, signal anchor, stop transfer. The type 4B has two signal anchors, and then from then on you go signal anchor, stop transfer, signal anchor, stop transfer, signal anchor, stop transfer. So then you can keep on going, and then the number of topogenic sequences will determine whether this carboxyl terminal domain is cytosolic or luminal. Okay, you can have different number of transmembrane domains. So from these things, the questions are going to be, if I remove the labels that say cytosolic or luminal, or remove the labels that say SI or STA and put charges here, you should be able to recognize the different types of proteins. Okay? And with these other ones, if I tell you something is a type 2 protein, if I say cadherin is a type 2 protein, you should be able to figure out what is the calcium binding domain of cadherin. Is it the N-terminal or the C-terminal? Because it's going to be the extracellular, the luminal side. That's going to be binding to calcium, so you should be able to figure things out like that. Aren't cells complex? All right, everyone get that? Hopefully. Okay, so that's membrane proteins. We're going to quickly now go into membrane transport. Okay, like I said, there's some good movies in Achieve, and there's other movies online. The stuff that you need to memorize, though, is sort of more, if I modify these, and if I add domains, and I ask you how many transmembrane domains are there, how many luminal domains are there, etc. All right, so let's go on to the last section, which I hope I can finish by the end of Wednesday. Okay, so we're talking about transport across membranes now. Membranes are semi-permeable, right? Things don't tend to cross. Gases cross, small uncharged molecules cross, but you need things to cross a membrane for cells to function. So there are different ways that you can get things across, and this is sort of a table overview, and we'll go through these sort of different processes. So simple diffusion isn't going to allow you to get proteins or ions across the membrane. You need large things in the cells, you need ions across the cells. So you need either facilitated transport, which we'll talk about today, or active transport and co-transport, which we'll talk about on Wednesday. Okay, so these are transports that's facilitated by membrane proteins. So you need proteins within the membrane to allow this sort of function. The function of the cell is dependent on having proteins in its membrane. Okay, so we're going to talk about different proteins that allow molecules to cross. So either through facilitated transport, which means going down the diffusion gradient, or a diffusion gradient, or active transport that involves pumps. Okay, so we'll be talking about channels and transporters and ATP-powered pumps. But just to kind of orient you to this sort of visual representation of things, what are these little things that it's trying to show you? In gray, you have the plasma membrane. Okay, so the plasma membrane is semi-permeable. You're not going to get ions across it or proteins or large molecules. So you need ways to get things through the protein. So the gray represents the plasma membrane that you need to get things through, not through the protein like I just said. I gotta talk slower. I'll slow down now. All right then. How's your weekend? Yeah, Liverpool lost a couple of games. That kind of stuck LFC. That was a bit depressing. Anyway, so you have a plasma membrane. You will see that there are gradients across plasma membranes, gradients of molecules. A lot of them are maintained by the cell. So these little triangles represent gradients of molecules. Okay, there's different gradients on one side of the plasma membrane versus the other side of the plasma membrane. So sometimes transport is down the gradient. So this is showing you there's a high gradient on one side and a low gradient on the other side. So sometimes there's two gradients, sometimes there's one gradient. So this little diagram is just showing you the plasma membrane, and it's trying to show you that there are gradients of molecules, and whether molecules are moving down a gradient or up a gradient. So it's either moving down its concentration gradient or against its concentration gradient. So that's what it's trying to show you. Okay, there's different types of proteins that allow facilitated transport or active transport, or a symporter, which is sort of a combination of two molecules. Sometimes things are moving against a gradient, sometimes things are moving with a gradient. And we're going to start off by talking about facilitated transport, where the driving force is a concentration gradient. So there's already a high concentration of molecules on one side of the plasma membrane, and this concentration gradient is going to drive movement from the high side to the low side. Okay, so this doesn't require ATP specifically. ATP was probably used previously to set up this gradient, and we'll talk about that. So there's high potassium outside of the cell because things happened previously. Okay, but now you have a concentration gradient, and molecules are going to move down this concentration gradient across that phospholipid bilayer, which is semi-permeable. So they can't go right through the membrane. They have to go through proteins that are called channels or gates or transporters. Okay, these are all very similar structures. These are just proteins that are going to allow specific molecules. They select what molecules can get through. So it's not like anything is going to get through. These proteins are specific. So you're moving these hydrophilic substances. You can move an ion, or you can move a protein down its concentration gradient because it's moving through a protein channel or a gate. It's not coming in contact with those hydrophobic tails of the fatty acid chains in the lipid bilayer. You're moving through proteins. So this movement is much faster than would occur by diffusion. Diffusion, you likely won't get ions across. This way you can. It's specific. Okay, so these things are transporting potassium, or they're transporting a specific amino acid, or transporting glucose. Okay? So it's very specific to what it can do. It's also saturable. What does that mean? That means it can only do it a certain rate. So later we'll talk about a GLUT transporter. So you're facilitating transporting of glucose from high concentration to low concentration inside the cell. But you can only go so fast because glucose has to bind and then release, bind and release. So you can saturate how fast things will go. Okay, so facilitated transport is faster than occurs by diffusion. It's specific to the molecule that's being transported, and it's saturable, so there's a limit to how fast it can go. Right? Okay, so we're going to look at specific types of things. So sometimes things are called channels, and sometimes things are called gates, and sometimes things are, well, less label here, transporters. So they're almost all the same thing. They're just proteins that allow things through that are specific. In general, something that's called a channel is just open all the time. So we'll talk about the potassium resting channel maybe in a second. So the channel is open and allows potassium to go through, but it's only going to allow potassium to go through, and it's going to be limiting how fast potassium can go through. It's not infinitely, it can't transfer everything. Okay, so channels tend to be open. You have other structures called gated channels, where they are open under certain circumstances. So you have molecules that are high concentration on one side and a low concentration on the other side, but if this channel is closed because of a gate, then things aren't going to move. So they're gated because they can be opened under different conditions. For example, there are voltage-gated channels. So a change in voltage is going to open up this channel, and then molecules can flow down the concentration gradient. So we've seen this type of transport without maybe necessarily calling it that, and that's the voltage-gated calcium release channel on the sarcoplasmic reticulum. If you remember, you have a T-tubule that's carrying this nerve impulse, which is basically voltage. That T-tubule is going to be right next to the sarcoplasmic reticulum, and it's going to release calcium from the sarcoplasmic reticulum. That calcium release channel is a voltage-gated channel. There's lots of calcium in the SR, no calcium in the cytosol. So it's going from a high concentration to a low concentration. So when you open up that SR-gated channel, the calcium is going to come out into the cytosol. It's going to go bind to troponin and then do its job. Meanwhile, there's another pump that we're going to talk about that's pumping it back into the SR quickly. Okay, so channels are open. Gated channels can be opened and closed via signaling molecules or voltage-gated channels, such as this example here. Okay, channels, even though they're open, they are specific, right? So as I already mentioned, the potassium resting channel. Okay, let's kind of look at how that works. So this is facilitated transport. This is transport going from high concentration to low concentration. It's specific, in this case, for potassium. And so you think, well, how can it be specific for potassium? Well, it's because it's set up so that potassium has the same energy in the channel as sort of out of the channel. Okay, so if you have high concentrations of potassium on one side of a plasma membrane, what is that potassium doing? Potassium is an ion. It's a positive ion. It's going to be surrounded by the shell of water. Water has a negative dipole, potassium is positive. So if you think about it, potassium on one side of the plasma membrane is going to be surrounded by water molecules. It's quite happy there. It's a neutral charge. So potassium is going to be surrounded by water. It's quite happy to be surrounded by water. It's a nice neutral charge. Okay? If you imagine a plasma membrane, you have high amounts of potassium on one side, low amounts of potassium on the other side. Potassium is on one side, it wants to get to the other side and wants to go down that concentration gradient, but it's happily binding with this oxygen in the water. Why would potassium break its bond with this oxygen to go down that concentration gradient? There's a certain energy amount that's needed to do that. So what the potassium resting channel does, it provides oxygen molecules such that potassium can let go of those water molecules and bind to oxygens that are present within the side chains of the amino acids. Okay? So the resting channel, this protein channel is made up of proteins, as I said. Those proteins have peptides, amino acids. Some of those amino acids have oxygen. So the oxygens that are present within the potassium resting channel, the spacing of them is identical to the spacing for potassium in water. So for potassium to let go of the water on one side of the membrane and bind to the oxygen in the resting channel is sort of neutral. It's happy to do that because it's the same energy. So potassium will release its water, go into the channel. It's driven by its concentration gradient. On the other side of the channel, there's more water, so potassium can bind to the water on that side. So in terms of energy, potassium is going to be bound to water, bound to oxygen, bound to water, and it's happy to use that concentration gradient of energy to do so, and it can do so because of that. If you consider other ions, such as sodium, sodium has the same charge, it's a positive ion, it too has a hydration shell, it too binds to water. So sodium that's on one side of the plasma membrane is bound to water. If sodium approaches the potassium resting channel, it needs to release its oxygen and then bind to the oxygen within the potassium resting channel. However, those oxygens are a different size. The sodium ion is different size than the potassium ion, it's smaller. So if sodium tries to enter that potassium resting channel, it cannot interact with the same number of oxygens. Here it's showing you interactions with 4 oxygens, which is sort of easy to imagine. So if sodium is bound to 4 waters, it wants to bind to 4 oxygens in the resting channel, it can't. So it does not release from the water. There's no reason for it to release in water. It's more favorably energetically for sodium to be bound to that water molecule. So sodium will not release from the water molecule because it can't really find favorable conditions to go through that resting channel. And this holds true for other ions. Other ions are either too big or too small, so none of them are going to go through this potassium resting channel. There are other channels for other ions. The potassium resting channel is specific for potassium. All cells have that because all cells have this charge gradient across them. Makes sense how it's selected for potassium? What can you do with that? So I'll describe this now, and then we'll start again here next time. Okay, so you have this little experimental setup where you have a chamber here, aqueous chamber, and you have a membrane present. So it's an artificial membrane, but it's a membrane present here. You dissolve salts on both sides. The charges are the same, so it's sodium chloride and potassium chloride on both sides, but you dissolve sodium and potassium chloride on both sides such that there's more potassium on one side versus the other side. So the number of negative and positive charges is the same, right? So when you look at the difference in charge, they're the same, but there's more potassium on one side. There's 150 millimole of potassium on one side and 15 millimole of potassium on the other side. So there's a concentration gradient. So potassium wants to go from this high concentration to low concentration. With a membrane, it can't. But if you put a potassium resting channel in this membrane, potassium can go down this concentration gradient. It can move from this high concentration to this low concentration. When it does, it takes that positive charge with it. Sodium, which is present, can't move through that channel. It just can't let go of those oxygens in the water. So potassium is going to go down its concentration gradient because it's going from high concentration to low concentration. And because of that, there's going to be a difference in charge across this membrane. And indeed, all our cells have this difference in charge across our membrane because of the potassium, whatever that molecule is, potassium resting channel present in all our cells. And we'll talk more about this next time.